In the name of God. So, what we are going to do today is to talk about immunology of infectious diseases. And this is a very important topic in immunology, uh, both for historical and practical reasons. Infectious disease immunology is indeed uh, a part of what we call infectious disease medicine. And infectious disease medicine is a very crucial part of medical sciences because one of the major challenges that human beings have had during history have been infectious diseases. In the introduction class, we talked about some major pandemics that had happened during the course of human history. We talked about bubonic plague. We talked about, I think, uh, typhus epidemics. We talked about other infectious diseases like cholera and specifically about smallpox, whose history is closely entwined with the history of immunology. And today, what we are going to do is to uh, talk about uh, factors which determine the type of immune response against infectious agents. So, what I'm going to do today is to give you a general overview. And this class is mostly sort of an undergraduate level class, but you guys are postgraduate students, so you need to read the textbook, the Abul Abbas textbook, or if any other textbooks that uh, you're going through. And I think it's chapter 13 in Abbas book. And there are much more details about immune response to infectious agents there. So what I'm going to give you today is basically an overview about immune response against infectious agents. So there are three types of factors or three sets of factors which influence our immune response against infectious agents. The type of the infectious agent, so here I have written type of the microbe, but it's better to call it type of the pathogen or type of the infectious agent because not all pathogens are necessarily microbial. So we have infectious agents like worms or other macroscopic entities which don't qualify as microbes. So the type of the pathogen or the type of the infectious agent. Am I dealing with a virus, with a bacterium, with a fungus, with the eukaryotic agent? So the type of the pathogen is going to be one of the determinants. The other factor is the target organ or organs. Are we dealing with a systemic infection, which is present in major like body size, including blood, for instance, or are we talking about an organ specific infection? So if I put these two factors together, I can talk about, say, viral infections of the respiratory system. The infectious agent could be a virus, the target organ or the target system could be a respiratory system. I can talk about bacterial infections of the CNS, bacterial infections of the central nervous system. I can talk about uh, bacterial infections which are systemic. Multiple organs are involved, and even bacteria could be retrieved from blood. And if a bacterium could be retrieved from blood, it means that it's everywhere. So these two sets of factors are very important. But what we are going to do in this class, and pretty much in immunology textbooks, that's the way that it's done, is to focus uh, on a third factor, which is the localization of the pathogen. And by localization, I don't mean anatomic localization. I mean localization at the cellular level, whether the pathogen that we're talking about is localized inside the cells, it's intracellular, or it's localized, or it lives outside the cells, extracellular. So we're going to use these factors, especially the third one, in order to understand and classify immune response. So what I'm going to do in the beginning is that to bring this factor onto the right side of the page, onto the left side of the page, and then show you four major groups of infectious agents that we have. We have bacteria, viruses, fungi, and parasites. And by parasite in here, I mean eukaryotic organisms, which can have a parasitic relation with us. So the word parasite is used in two senses. In a broad sense, all of the infectious agents have a parasitic relation with our body. So even viruses, bacteria, fungi, when they infect our body, they have a parasitic relation. But by parasite in here, I don't mean parasite in that broad sense. I mean specifically eukaryotic organisms like Giardia, for example, which is a single cell eukaryotic organism which causes giardiosis, or helminths, for example, worms, which are multicellular organisms. So we have these four groups of pathogens. I have deliberately excluded one important group of pathogens from this table. Do we have any pathogens that are not included in here? Something which is neither a bacterium, nor a virus, nor a fungus, nor a eukaryotic organism. Do we have any disease which is caused by something which is not included in here? We have one very peculiar group of pathogens which are neither bacteria nor virus, fungus, or parasites. They are prions. So prions are a specific group of pathogens which have their own group. They cause diseases like mad cow disease, for instance. And these are small, misfolded proteins which can infect our bodies and then cause misfolding, change the configuration of proteins in our body, including in our central nervous system. So I have excluded prions, and immune response against prions is a type of, is a little different from the immune response, if we can really call it immune response, compared to what we have against other pathogens. So we don't have prions in here, and we're not going to discuss them. Maybe in advanced immunology classes, we have a time to go through what our immune response could be against things like spongiform encephalitis caused by prions or mad cow disease or things like that, but we don't have them in here. So what I'm going to do is to put the two major localizations, extracellular and intracellular, in here, and then four major groups of pathogens in here, and then come up with some examples in the beginning to fill up this table. So for bacteria, the majority of bacteria live outside of the cells, like the E. coli, which causes diarrhea, or pneumococci, which can cause pneumonia. But we do have some bacteria which live inside the cells. They are intracellular bacteria. Can you give me any examples? Exactly. Mycobacteria are one major group of bacteria, but one major group of pathogens that live inside the cells. And we have two major mycobacteria which cause human disease: Mycobacterium tuberculosis and Mycobacterium leprosi, which causes exactly Mycobacterium leprae, sorry, which causes leprosy. So we have extracellular and intracellular bacteria. With regard to viruses, well, viruses always have to live inside cells because they don't have sufficient tools to produce their own proteins and generate their offspring. So they have to live inside cells. They are intracellular pathogens. For fungi or fungi, both pronunciations are correct. The majority of fungi are extracellular, like the candida or like dermatophytes, which cause a skin infection. But we do have some examples which live inside cells. We have a fungus named histoplasma, which causes a lung disease named histoplasmosis, which is very similar to tuberculosis, which lives inside the cells. How about eukaryotic organisms? Well, worms or helminths, they're outside cells. They're too big to live inside cells. But we do have things like plasmodium, which is the infectious agent causing malaria, which lives inside cells. So these are some examples of extracellular and intracellular pathogens. So I'm going to use this table in order to classify immune responses. But before doing that, I need to make this table a bit more complete, because not all extracellular spaces are like each other. We have compartments in extracellular space, and we do have compartments inside the cells as well. So what I'm going to do is to take a mycobacterium and a virus and show you in a very schematic way how they might differ in terms of the intracellular compartment that they localize to. On the left side, imagine that I have some mycobacteria, Mycobacterium tuberculosis. Mycobacterium tuberculosis enters the lung. If I inhale the droplets or like the aerosol that is produced by exhalations of an infected person, they go inside my lungs. There we have some sort of macrophages which are called alveolar macrophages. They are present in lungs alveoli, and those macrophages would phagocytose the mycobacteria. So something is produced in here which is called a phagosome, and phagosomes are usually merged with an intracellular organelle which is named lysosome, generating phagolysosomes. And a lot of pathogens are very sensitive to what is present inside lysosomes, and they die. But for mycobacteria, this is a safe environment. They have developed ways to protect themselves against lysosomal enzymes and the acidic pH and the rest of the things. So they live in here, they proliferate. After some time, they kill this macrophage. They come out, they get phagocytosed by another macrophage, and then the cycle is repeated and repeated until a lot of damage is caused in the alveoli, and something appears in the lung which is called mycobacterial tubercles, hence the name tuberculosis. This is an example of an intracellular pathogen.
Lung itself is the if someone is immunocompromised, doesn't have angular effects, it's possible for them to cross.
That's a very interesting question. So if I don't have the target cell, which is the macrophage, what would happen to that person? Well, in reality, we don't have a situation that the person is completely devoid of the so-called macrophage or monocyte macrophages. We do have situations in which macrophages are unable to kill the pathogen. Like we have a disease named chronic granulomatosis disease, or CGD, in which you have deficiencies in terms of like the intracellular killing mechanisms. But a disease in which we are completely devoid of macrophages, I don't know of such a disease. Nonetheless, if you imagine a scenario where you don't have macrophages and the person has inhaled mycobacteria, I guess what would happen is that those mycobacteria will be able to find another host cells for themselves. But I don't have precise information about that. So that's an interesting, even though it might be an imaginary scenario, but it's worth thinking. If I get rid of macrophages in the lung and then I have mycobacteria, do they really have any place to proliferate? I guess some epithelial cells would be the second preferable host for the mycobacteria.
They have similar constituents.
Yeah, the mycobacteria prefer to be basically engulfed by monocytoid cells and proliferate there. But if you don't have them, they can also proliferate in some non-preferable host cells. But that's a question that needs further study. So that was a good question, by the way. This is an example of an intracellular pathogen. On the right side, I have some viruses, again, schematic figures, like imagine these are hepatitis virus, hepatitis A or hepatitis B. And here I have a hepatocyte, a liver cell. The way that it happens is that they go and attack the liver cell and they go inside and start reproducing, producing their proteins and the rest of the molecules that they need. After some time, this cell dies, they come out, they go and infect another cell, and after some time, enough damage is caused to the liver, and we develop disease which is named hepatitis, viral hepatitis. This is another example of an intracellular pathogen. But these two intracellular pathogens are different in terms of the compartment to which they localize. This one is an intraphagosomal pathogen. It's inside vesicles. It's not in direct contact with the cytoplasm. It's inside the phagosomes. And this one is an intracytoplasmic pathogen, or an intracytosolic pathogen. And for immune system, it's very important. If you are an intracellular pathogen, are you going to localize inside phagosomes and proliferate there? Or are you going to be cytoplasmic? Because that would determine the next step in immune response. So what I'm going to do in here is that I'm going to put, this is intravesicular, this is intracytosolic, a line here and divide this compartment to two subspaces and then put pathogens there. For the extracellular, again, we have two major compartments. Sometimes when we say extracellular, it can be at the surface of epithelium, like a lot of pathogens that live at the surface of the skin, or at the surface of other epithelial layers that we have in our body. They do not penetrate the epithelium, they live there. But sometimes pathogens penetrate the epithelium and go inside the interstitial space. And from there, they can have access to blood or lymph. So they are inside blood, not inside blood cells, inside the fluid part of the blood, inside the serum or plasma. So these are two major compartments of the extracellular space. And if I want to sort of populate this space again, mycobacteria would be in here. They are intracellular, intravesicular pathogens, but we do have some bacteria like these two guys, which live inside the cytoplasm. Rickettsia is one of the pathogens that can cause diseases like typhus, the one that we talked about, Napoleon's army and their invasion against Russia and the rest of the things. And then we have chlamydia, which can cause sexually transmitted diseases, but it can also cause eye infection. One major eye infection that was once very common in Middle East is called trachoma, chlamydia trachomatis, which causes eye infection that can lead to eye damage and even blindness. How about viruses? Viruses are usually inside cytoplasms, even though they might get phagocytosed briefly sometimes and be present inside vesicles, but they get out and live inside the cytoplasm because that's where all of the required elements for them exist. For fungi, well, we don't have many intracellular fungi, but the histoplasma, if it's intracellular, it's usually circumscribed inside vesicles. Another fungus which you see infections with it in immunocompromised patients is pneumocystis. I don't know of any specific fungi which live inside cytoplasm, but my information might not be complete, so you can look up to see whether there is anything that lives inside cytoplasm. How about eukaryotic organisms? Well, something like leishmania, which can cause cutaneous or visceral leishmaniasis, lives inside vesicles, but for plasmodium, it's usually inside cytoplasm. How about bacteria? How about extracellular space? Well, it's very diverse. E. coli could live at the surface of the intestinal epithelium. So it's over epithelium, lives at the surface of epithelium, but it can penetrate the epithelium, get underneath epithelium and cause diseases there. We can have streptococci at the surface of epithelium, but they can penetrate and cause infections there. Some other bacteria like Neisseria can be at the surface of epithelium, like the one that causes gonorrhea, Neisseria that causes gonorrhea. So that's at the surface of epithelium, but it can penetrate and go inside the cells. Or Neisseria meningitidis, which can cause meningitis. It lives in the interstitial space. For fungi, usually at the surface of epithelium, but you can have them inside blood or interstitial space sometimes. And for the rest of the parasites, a lot of worms that we have live at the surface of epithelium, but some of these worms can be living inside the interstitial space as well. So this table is not complete. I have only some examples, major pathogens, so you can look up to see whether, for instance, sometimes we have fungemia, the presence of fungi inside the blood, or do we have any fungi that can live inside the cytoplasm? That's possible, but I don't have any examples in here. So why did I go through this? Because I want to classify immune responses based on these lines that we have in here. I have 4 spaces in here, 1, 2, say 3 and 4. And the way that I want to classify it in the beginning is to use this line in the middle and use it as a sort of a classifier for adaptive immune responses. Let's start from there. If you recall, in the beginning, in the first class, we talked about what is called the model of the immune system. Can you tell me what the model was? And then I drew something like this. I said this is a model of the immune system. Could you tell me what the model was? Do you recall? I asked you guys to write it down even on your pieces of paper. Actually, take a piece of paper and write it down. So scientific model, write it down. Take a piece of paper and pen, write it down. I want to see, have you sort of memorized that expression that I use? Well, science should be based on understanding and deduction and stuff like that, but sometimes it's good to memorize things. I gave you a brief explanation of what a scientific model is. It was a very small phrase. Write it down. Write it down. And then we said that we have physical models, we have conceptual models, we have mathematical models, and then this is a model of the immune system. What was it? Well, you can look up definitions in Wikipedia, but what is a model? Scientific model. Anybody?
A model is
A simplified representation of reality. A simplified representation of reality. The scientific model is a simplified representation of reality. All right. Why do we need a simplified representation? Because reality is very complex. If you want to sort of make a representation of reality which exactly matches the reality, you have to have a lot of information, which we usually don't. And then that representation, even if it's possible, might be difficult for our brains to analyze and understand. So that is why we go for simplified representations of reality. Simplified, because our information about reality is limited and our brain's processing power to understand is limited. So we go for simplified representations. And then we have physical models, like this model of the heart. Do you recall these things? Anything? Any recollection of what I said about models in the first class? Did I show you these things? I'm sure that I did, because that's what I do. Conceptual models and mathematical models. And then I said, what kind of model is this? We have immune system, two branches, innate immune system and adaptive immune system. The innate part is physical chemical barriers, comprises physical chemical barriers, and we have cells. And then the adaptive part, we have the part which is present in body fluids, humoral immunity, generated by B cells, and the part which requires cells in order to be active. And that is the cellular immunity. This is a model of the immune system. What kind of model is this? Is it a physical model, a conceptual model, or a mathematical model? This is a conceptual model. This is not physical. I haven't made anything. I have just made a concept map. I have drawn the concepts that I have in my mind, or I have extracted from immunology books in here. And this is not mathematical because there are no mathematical expressions. This is a conceptual model. And models, the important quote about models, write it down, write it down. What is the important quote about models?
all models are wrong, but some are useful.
Write it down. All models are wrong, but some of them are useful. What do we mean by wrong in here? They are just approximations of reality. This is a famous quote from George Box. Very important. So in science, we have realities, complex realities, especially in biology. The realities are very complex. We make simplified representations of those realities for educational purposes and also for better understanding because of the limited ability of our brains to understand. But we should keep in mind that these are not equal to reality. When we say simplified representation, it means it's an approximation. It's an approximation. Okay. So what is the how many Arabic speaking people we have in this class except Mr. Rezaya? How many Arabic speaking people we have in this class? Okay. What is the word for approximation in Arabic? Okay. Oh shoot! No interns. Okay. So that's very important. It's a very fundamental concept. It's a core concept in science. And most people are either not familiar with this concept or kind of ignore it. So I'm attached to this. I cannot see it. Okay, it's a tagrib. It's an approximation. It's not. It's just trying to make something similar or more, yeah, understandable, sort of. Okay, this is a conceptual model. So, and then in this model, we had innate immune system, adaptive immune system. What I'm going to do in this class, is to try to develop a model for our immune response against infectious agents. What I'm showing you in here, this is a model. of the different spaces that these cells can be, or these pathogens can be. So what I'm going to do in here is to sort of use this model to build up, use to this table to build up the model that I want to make. And the way that I want to do it, we have an innate immune response and adaptive immune response. For innate immune response, we have barriers. So this epithelium is a barrier, but when it's penetrated, then that barrier doesn't really matter. So for epithelial ones, we have a barrier, tries to keep them in place, but when that's penetrated, well, those pathogens have access to the rest of the body. Then we have phagocytes. How about phagocytes? Well, phagocytes usually don't have access to things which are at the surface of the epithelium, because the phagocytes don't get through the epithelium to the surface. But phagocytes can have access to the rest of the things in here. They can have access to things which are in the interstitial space or blood. Phagocytes are there. Vesicular, well, this is inside the phagocyte. It's phagocytose something. It's inside the phagocyte. It tries to kill it. It might be successful in killing or it might be unsuccessful in killing. How about things which are hiding inside the cytoplasm? Well, if it's inside the cytoplasm, it cannot phagocytose it directly, but it can phagocyte the entire cell, perhaps. Or it can wait for the cell to die and then phagocytose the pathogen. So that's another way. So basically, we can say except this space, epithelial region, phagocytes can have access to the rest of these spaces. The problem is for B lymphocytes and T lymphocytes. And I want to give this question to you guys. Imagine that this line in the middle is a line which wants to divide the responsibilities between humoral and cellular immunity, B cells and T cells. We have extracellular space and intracellular space. So this is a basic question, logical question. If you guys want to divide the responsibility of defending against pathogens between B cells and T cells, give one region to B cells to defend and another region to T cells to defend. One space to B cells to defend and another space to T cells to defend. Which space would you give to B cells to defend? And which space would you give to T cells to defend? And why? And why? Let me ask you. You give extracellular to? Why do you give extracellular space to B cells and intracellular space to T cells? I want to give intracellular space to B cells. Why? Because it's written in the book? You're right. So the way that it works is that extracellular is the B cell domain. Intracellular is the T cell domain. But why? Why?
T cell, intracellular, cellular immunity.
So the way that you're explaining it is again the textbook answer. The textbook has said that humoral immunity works in the extracellular space better, but if something hides inside the cells, I would give it to T cells. But I want sort of the logic behind that. Imagine that you didn't have the textbook. What you would say? Too complicated. This is a good answer, but it's too complicated. Make it simpler. Why? I better not give the intracellular space to B cells to defend. I want this region to be defended by B cells. What's wrong with that? The problem is that antibodies don't go inside cells. Antibodies, B cells, how do B cells work? They produce antibodies. Antibodies can go inside, can go everywhere in the extracellular space, but they do not penetrate the cell membrane. At least classical antibodies don't do that. So if something is hiding inside the cell, if it's a virus or something is inside the cell, antibodies cannot have access to that compartment. So for that reason, you need to look for another solution. Antibodies cannot go inside the cells. So if I want to look at this very simplistically, We're making a model. So this is a very simple model. Very simplistically, I would give this region to B cells to defend because antibodies can't be present and go everywhere in the extracellular space. But I would give this region to some other cells, maybe T cells. All right. Okay. So based on this, I want to give you a sort of a puzzle to solve. Have you guys been vaccinated for hepatitis B? Hepatitis B, Hep B shot. All right. So if I get vaccinated for hepatitis B, which is usually a 3-dose vaccine being injected at days 0, 30 days, and then 180 at the end of the 6 month. If I want to check whether the vaccine has been effective, because some people get vaccinated, but they don't get protected. They don't respond to the vaccine. And especially in the Middle East, this is very common. This is relatively common. I should go to a medical laboratory and get tested for something. If that something is present in my blood, that means that I'm protected. If that something is not present in my blood, it means that I'm not protected. What is that something, Mr. Rezai? I would be checking my blood for the presence of antibodies against hepatitis B virus. If I have a high concentration of the antibody against hepatitis B virus, it means that the vaccine has been effective. If I don't have sufficient levels of antibodies, it means that the vaccine has not been effective. All right. So by looking at this table and what we just said, we said that antibodies are effective in the extracellular space. Viruses are inside cells. That's one piece of fact, assuming that is correct. And the other fact is that when I get vaccinated for hepatitis B, I go and check my cells for antibodies. I see a contradiction in here. What is the contradiction? The contradiction is that I said viruses are inside cells. Antibodies don't have access to them. Nonetheless, when I get myself vaccinated, I go and check for antibodies. So something is wrong in here. Do you get the contradiction or not? Yeah. Do you get the contradiction? What is this paradoxical situation? I said viruses are inside cells. Antibodies do not go inside cells. So T cells should take care of that. But when I go and vaccinate myself for a viral infection, for hepatitis B, for SARS-CoV-2, for mumps, measles, rubella, all of the time, I would go and check myself for the production of antibodies. What's wrong with the model? What's wrong with the model? Okay, I would say every model is incomplete. So this model is incomplete. All right, so that is exactly my question. What is the detail that needs to be added to this model to solve the problem? Viruses, antibodies are against the viral proteins. For instance, for hepatitis B, we have an HBs antigen. Antibody is against that. So if I have a virus, antibody should go and bind to the surface of the virus. So no, it's protein of the virus. It's the protein of the virus. So when we talk about hepatitis B vaccine, we have an antigen called HBs antigen. HBs antigen. HBs antigen is what is present in hepatitis B vaccines. So you inject people with hepatitis B with HBs antigen, and they produce antibodies against the HBs antigen. So when you have antibodies against HBs, it would go and bind to these things in here. HBs in here is shown with this HBs antigen. Well, these are apparently different domains. Is present at the surface of the virus. Antibody would bind to that. But if it's inside cells, you don't get any antibodies inside the cells. That's the problem. Yeah. Anybody else? Any answers?
Because the immune response is not one-sided. It's not only one side. It's integrated. It's connected to each other. So you can't just give a side to a different cell. You can't give intracellular only to T cells because T cells also have a protein.
For now, imagine that, okay, yeah, B cells and T cells always work together. I told you the model that is correct, better than the current model that we have is a network model. B cells and T cells always collaborate. But still, I cannot solve this, despite the fact that my model is not complete. I would say, let's make it simpler. Viruses are located inside the cells. All right? Antibodies cannot go inside the cells. Nonetheless, when I get myself vaccinated, I check myself for antibodies. There is something wrong in here. Exactly, that is the answer to the question. What I'm showing you in here is the final destination of the pathogen. But for the virus to reach the final destination, it has to go through a path. I said that viruses are inside cells. Okay, when the infection is established, they are inside the cells. But for the establishment of the infection, virus has to go through other spaces in order to reach there. So if I look at it just this way, well, antibodies cannot have access to what is inside the cells. But all of these pathogens have to go through some paths. And for intracellular pathogens, they always have to go through extracellular spaces before they reach their final destination. So, for instance, for hepatitis B, what would be the path? Hepatitis B could be sexually transmitted, could be blood transfusion, could be related to blood transfusion. So if it's sexually transmitted, in the beginning, it's at the surface of the epithelium. Then it penetrates the epithelium, enters interstitial space, and then from there it goes to blood. Then it circulates inside the blood until it reaches a hepatocyte and goes inside the hepatocyte. So if I want to consider this 1, 2, 3, 4, it has to go through space 1, subspace 1, then subspace 2, then it reaches subspace 4. If it's blood transmitted, it's in the beginning. It appears in the blood in the beginning, but because it's transfused, it circulates in the blood, and then finally it gets to hepatocytes and becomes intracellular. So here I have drawn some examples of pathogens' paths. The path that the pathogen needs to go through in order to establish its infection. And the way that I made this, like these sort of curved arrows that I'm showing you in here, I have made it like this. This part is the route of entry, where it enters. This is the path, and this is the final destination. This way, okay? So we have some pathogens like this. We have some pathogens that start at the surface of the epithelium, they remain at the surface of the epithelium, they proliferate at the surface of epithelium, they cause disease at the surface of epithelium. They do not go anywhere else. Can you give me an example? Write it down.
E. coli.
E. coli is a good example. A lot of E. coli's, they enter the body through like contaminated food or water. They go at the surface of intestinal epithelium. They stay there. They produce toxins, for example. Giardia is another example. They do not penetrate. Giardia is another example. Candida is another example. Very good. A lot of fungal infections of the skin, they are always at the surface of the skin. They do not penetrate. Helicobacter pylori, H. pylori, it's at the surface of stomach epithelium. It remains there. Usually it doesn't penetrate. So these are epithelial surface pathogens. All right. Sometimes it's like this. At the beginning, it's at the surface of epithelium, but then it penetrates. E. coli has another version which is called entro-invasive E. coli. In the beginning, it's at the surface of epithelium, but then it penetrates and goes sub-epithelial. So that's one example. No, because malaria, the final destination is inside the cytoplasm. So what else? Some fungi, in the beginning, they are at the surface of epithelium, but then they can cause fungemia, especially in immunocompromised people. They go inside blood. So we have examples for this. How about this one? In the beginning, it's at the surface of epithelium, then it penetrates, then it gets phagocytosed and goes inside vesicles and remains there. Mycobacterium tuberculosis, we can say that, sure, at the surface epithelium. Then it's either directly gets phagocytosed by alveolar macrophages, or it goes through an interstitial stage, sure. How about this one? This is very important. First at the surface of epithelium, then it penetrates. Then it goes and finds a target cell and becomes intracytosolic. All viruses, all viruses that are either sexually transmitted or they have fecal oral route of infection are like this. Fecal oral route of infection like rotavirus, like poliovirus. They enter the body through contaminated water or food. In the beginning, they are at the surface of intestinal epithelium. Then they penetrate and go interstitial. Then they enter blood. Then they go inside the blood and look for the cell for which they have tropism. Remember this one. Tropism. Tropism. Like for poliovirus, it's neurons in the anterior horn of the spinal cord, motor neurons. For hepatitis virus, it's hepatocytes. So it goes inside cytoplasm.
Like that.
The Black Death, it was not a viral disease. It was plague, Yersinia pestis. Yersinia pestis. Actually, that's a good one because now I'm going to show you this. Sometimes the pathogen skips the epithelial process, epithelial barrier. It enters blood directly. How is this possible? Well, if it's through insect bites. Insect bites. So Yersinia pestis is a good example. It's through Yersinia pestis for plague. No, not malaria. Don't get malaria, plasmodium, it goes inside cytoplasm. So this is something that enters the blood or interstitial space and remains there, proliferates there and remains there. Yersinia pestis is kind of one example. We have other pathogens too. I will let you guys look it up. So insect bites, sometimes, especially if it's a bacterial infection, it usually remains in the interstitial space. How about this one? It goes inside vesicles and remains there. But in the beginning, it's interstitial. Do we have something like this? Leishmania, that is transmitted by mosquito bites. It's interstitial in the beginning. And then it gets phagocytosed by macrophages and remains there and causes disease. And finally, this one. Well, all viruses which are blood-borne viruses. And malaria is in here too. Exactly. So insect bite, and then it gets inside the cytoplasm of hepatocytes and then red blood cells. So I'm going to let you guys contemplate on this. This is a very simple version of the paths that pathogens take. And it doesn't show everything. Again, it's a model. It's a simplified version of reality. Why it's simplified? Because a lot of pathogens move between these compartments, especially viruses. Go back and forth. Viruses, once they are inside cytoplasm, they don't remain inside cytoplasm forever. They kill the cell in which they are proliferating. They get outside, they come back to blood, and then they infect cells again. And they come back to blood or blood plasma and they infect again. So sometimes they go back and forth. So, but one thing is obvious from what I'm showing you in here. All these pathogens have something in common. They always start their journey from extracellular space. We don't have anything which is directly inside the cells from the get-go. It's kind of impossible unless you inject infected cells from one person to another person. Okay, so for transplantation, it's different. So if you have an infected transplant, like if I have a transplant which is infected by CMV virus or EBV virus, and then you transplant it, that way you're taking cells which are already infected to the body of the person. So that's an exception. That is something which does not happen naturally. Transplants don't happen naturally. So what we're talking about in here is about the natural course of infection. And if it's a natural course of infection, it always starts from here. It has one very major implication for the immune system. What is the major implication? Pathogens are always exposed to antibodies, which are present in the extracellular space, even though their final destination might be inside cells. But before they get to the intracellular compartment, B cells always have a chance to defend. Their defense might not be successful, but they do have a chance to defend. All right. That is why when I get myself vaccinated for hepatitis B, I check myself for antibodies, because antibodies should be produced and they should be present in the extracellular space. And if I get a needle stick, or if I get transfused with contaminated blood, or if I have unsafe sexual behavior, those antibodies are present in the extracellular space and they try to block this path in here. If their defense is not successful and the virus goes inside, then you have to come up with other solutions like T cells. But other than that, you have it in here.
Always like sure when even let's say the vaccine is successful inside a person, they ended up taking, for example, transfused blood with the virus. Is it a sure way that they do not get infected with it?
No. Vaccines don't have 100% efficiency. If that's what you mean. When we get someone vaccinated, assuming that the vaccine has been effective, that effectiveness is not necessarily 100%. Well, yeah. So if you are vaccinated, if you have good antibody titers, if you get a lot of viruses inside your body, it's possible that some of those viruses might be sort of skip through the humoral immunity and then infect the cells. So what I'm going to do in here is to, because that's already 45 minutes. So let me just stop recording in here and save this file so that I can upload it easier onto the YouTube channel, and then we will continue with the class.